All right, here we go. This is part 2 of my series going through the Endogenous Retrovirus Handbook. For those interested, that would be this book right here. Again, the Endogenous Retrovirus Handbook, Dismantling the Best Evidence for Common Descent. And so in this series, what I am doing is going through every single chapter. It's basically a walkthrough. And so rather than just a strict audio book, I will be reading through again each chapter. But at times I may elaborate on certain points. I will give some commentary. And I will, especially at the end of each episode, again, this is part 2, I will provide more details. I will update the audience on the research I've been doing in terms of model building and answering common objections, challenges, and criticisms to this specific book. Now, this is a Patron-first series. So what that means is each part will be put out to Patreon first, and therefore the patrons and the ministry partners, they will receive first access to each one of these episodes. After about a week or two, I will upload it publicly in order that everybody can benefit. I do believe it's highly important that everybody watches this series, studies this series, because the evolutionary community promotes, they advance endogenous retroviruses as the single best evidence for common descent. Many will argue that it is a line of evidence that makes common descent or evolution undeniable. And therefore, I want my brothers and sisters, I want the body of Christ to be well equipped and ready to demolish the critics when they bring up arguments like endogenous retroviruses. I also want the subscribers, supporters, and patrons to be able to follow along as I go through each chapter. And so if you do have a book yourself, that's awesome, but I do want to make sure that you all have a PDF copy as well. And therefore, please reach out, send me an email or message me on Patreon, and I will make sure to send you a free PDF. Now, since this is part 2, I am going to just get right into the next chapter, which is the introduction. So part 1, make sure to check it out. It is in the description box. That was the preface. It was about an hour because it was the first part in this series. I went over a lot of the basics, a lot of the necessary information on how I am going to put out each part, what we're going to focus on. I went through the website as well, showing people where they can find more content on endogenous retroviruses, and also where they can purchase the book, the Endogenous Retrovirus Handbook. And so please do make sure you watch part 1. Again, that was the preface. I also offered about 20 to 25 minutes worth of very important commentary. And there will be commentary as well in the introduction. But as we move into the actual heavy material of the book, in terms of the chapters, which in part 1, I went through the table of contents and I gave a brief overview of what I touch on in each chapter. And so I think that's important for people to watch as well. And so again, please make sure to share this content around, and hopefully you can get a lot out of it, because again, this is a big subject. It's a technical subject, and I want to be as comprehensive as possible. And I figured to be comprehensive, a walkthrough of the book itself would be beneficial, I think, to the audience. And so with that, we are just going to get right into the introduction section of this book. And we are going to start right from the top of this section. Again, introduction. Part 1 was the preface. Endogenous retroviruses are small pieces of DNA that are found within the genomes of many kinds of creatures. How we can recognize an ERV element will be discussed thoroughly in Chapter 1 of this handbook. According to the standard evolutionary story of endogenous retroviruses, millions of years worth of evolutionary history has resulted in the incorporation of retroviruses into the genomes of primates and other lineages. On occasion, these retroviral insertions have managed, as the story goes, to invade through the germline. As a consequence of this, they have been passed on to descendants. Essentially, these retroviruses are passed on vertically rather than horizontally. To the evolutionary community, endogenous retroviruses represent viral fossils. We find these retroviral-like sequences in the same locations within the genomes of different organisms, humans and chimpanzees, for example. They fall into a nested hierarchical distribution where more ERV-like sequences are found in similar locations between a human and a chimpanzee, and with fewer ERV-like sequences shared between what is assumed to be a more distant relative, such as a gorilla or orangutan. Evolutionary scientists appeal to what is called a family tree that they interpret as being the result of common descent. Now, I want to provide a little commentary here. When it comes to the hierarchical patterns, I touched on in part 1 the fact that if the majority of ERV-like sequences, now I say ERV-like sequences because over 90% of what the secular or the conventional community or literature consider ERV sequences actually represent solo LTRs. An LTR is a long terminal repeat. The structure of an ERV, which I go into detail in Chapter 1, the important details it's called, they comprise, component-wise, LTRs on either side. They composed GAG, POL, and ENV genes. The ENV gene is the envelope protein of the retrovirus. Now, again, over 90% of what evolutionists consider to be ERV sequences represent solo LTRs. In part 1, I briefly went over why these so-called degenerated ERVs that only comprise the solo LTRs probably represent functional stretches of DNA rather than being degenerated ERVs. And so the point I'm trying to make here, in light of nested hierarchies, is that if the majority of ERVs and ERV-like sequences are created units of DNA function, then it's no surprise that humans would share more ERVs and ERV-like sequences with chimpanzees than we would with a fish or with a dog. This should be obvious. It's because in terms of morphology and in terms of anatomy and physiology, humans share more with a chimpanzee. Humans share more with a bonobo or an orangutan or a gorilla than they do with a bear or a dog or a fish. And so from the design perspective, this nested hierarchy is a side effect or a consequence of God creating organisms in the beginning. By definition, humans would have to be more similar to one creature, let's say the chimpanzee, than to another, in the same way that human engineers, when they design vehicles, a sedan is going to have to be more similar to another mode of transportation, like a hatchback, than a sedan would be to another mode of transportation, like a truck or a tractor trailer or an airplane. Human engineers accidentally build and design things that fall out into hierarchical patterns. Human designers and human engineers are not sitting back and planning out a process of design that would result in hierarchical patterns. No, this is happening just by chance. It's the nature of design. And therefore, if God designed the biological world would naturally follow in hierarchical patterns where some creatures will be more similar to other creatures, and those same creatures will be more dissimilar to other creatures. And so that's why I oftentimes point out that the important question when it comes to this debate of endogenous retroviruses is this: are endogenous retroviruses really the ancient remnants of past viral infections, or are they created units of DNA function? And so let's get back to the introduction. They interpret the nested hierarchical patterns within primate genomes in terms of their arrangement of these ERV-like sequences as reflecting common ancestry. Proponents of evolution would say all primates are related, while biblical creationists would say there exists separate ancestries. Evolutionists would say the common ancestry model better explains the data than the separate ancestry model does. Is an appeal to common ancestry the only way to explain the existence of endogenous retroviruses and their pattern of distribution found within primate genomes that end up being represented on family trees? Or is there a superior model that better explains the entirety of the data associated with these interesting DNA elements? That is exactly what this handbook will answer. The question of ancestry is an incredibly important subject, and the objective reader will have no choice but to reject common descent and accept separate ancestry. The model that I will be putting forth throughout this book predicts that these DNA sequences, known as ERVs and ERV-like elements, exist for functional reasons. If these various classes of DNA units are functional and beneficial to organisms, with ERV-like sequences being found in the genomes of organisms they are found in, then we can rightly conclude that these are found where they are for a reason and for a purpose. I offer numerous technical papers directly from the primary scientific literature that firmly validates and backs up the arguments I am making throughout this valuable handbook. I argue these DNA elements are not the ancient remnants of past viral infections, but are instead designed units of DNA function. I'll provide a little commentary here. When you get to the final chapter, I provide technical paper after technical paper after technical paper, literally dozens of technical papers directly from the conventional literature, that there should be no doubt that what we're looking at in terms of ERVs and ERV-like sequences are created units of DNA function. You are going to find throughout this series and just through reading the book that ERVs are not just functional on a basic level. No, they are literally necessary to sustain healthy life processes in the cell. They help to determine cell types. They act in gene expression, gene regulation. They function in embryological development. They function in the immune system. They function in cell stress responses. They act in viral mimicry. It is function after function after function. And not once has an evolutionist, including the PhD evolutionist, provided any empirical data for how these endogenous retroviruses could evolve these functional abilities that without these functional capabilities of the ERV, we wouldn't exist. Mammals wouldn't exist. Life wouldn't exist. Why do humans and chimpanzees share endogenous retroviruses? Well, why do sedans made by GM and hatchbacks made by Ford share engines. It's because the engines are necessary for the vehicle to function and for the vehicle to do its job, to take a person from one place to another. ERV sequences that are necessary for life to exist because they function in embryological development, or they function in safeguarding the growing baby inside the mother's womb from harmful viruses, then we share those ERV sequences because we need them. We require them. We require them. The chimpanzee requires them. This is good design. This is evidence for common design. Now, if they were junk, on the other hand, and they were useless and they had no important functions, then these may provide the common descent model and evolutionary proponents the kind of evidence that they so desire. Nested hierarchies in general. The shared features that we see in the biological world allow us to do what? Well, they allow us to study a creature and generalize our findings to understand the biological world. The fact that we are designed, or that the biological world is designed in a logical, orderly manner, helps us as humans and helps scientists study the biological world and understand God's design. If there was no order and all there was chaos with no patterns at all, then we would not be able to make and uncover the kinds of discoveries that we have done. The biological world is designed in nested hierarchical patterns for discovery purposes. Life is designed in a way that allows us as humans to study the biological world and make some incredible discoveries. Again, if the created order was chaos and was not designed in hierarchical patterns, discoveries in biology would be very difficult. Therefore, we have an elegant and a sophisticated reason for why God created hierarchically. The way God designed the biological world allows for what? It allows for discoverability. And this screams of design. In the second chapter, I go through design diversity, and I in great detail. In chapter 3, I go into the mainly agnostic lines of evidence for common descent. That chapter is called the non-discriminatory evidence. And so I talk about hierarchies in great detail. Okay, so let's continue here. I also deliver a comprehensive explanation for why these created units of DNA function are found in similar or corresponding positions in the genomes of various organisms, and why these sequences, including their mutations, fall into nested hierarchical patterns. The reader will also get a meticulous explanation that is supported by primary source data for why these DNA sequences resemble that of viral genetic material. Advocates of common descent particularly like to concentrate on the ERV sequences that are found present in the genomes of humans and the great apes, such as chimpanzees, bonobos, gorillas, and orangutans. Evolutionists would regard humans as part of this family. The similarity in sequence patterns mentioned earlier in this introduction, as well as their presence in corresponding positions within the genomes of these various creatures, will be explained in great detail. By the end of this book, the reader will have been confronted with overwhelming evidence that both refutes the common descent model and confirms separate ancestry. The separate ancestry model would propose that humans are not related to any other form of life, and this includes the great apes. If aspects of this introduction are already unclear to some readers, there is nothing to worry about, as I have written chapter 1 of this handbook specifically with the intention to provide the reader with all the significant and essential details to fully understand what exactly endogenous retroviruses are, and why the evolutionary community looks to these fascinating genetic sequences as evidence for common descent. Chapter 2 very thoroughly covers the design diversity model to set the stage for the all-out dismantling of ERVs and the entire junk DNA paradigm as evidence for common descent. Now, let me provide a little more commentary here. Why refuting the junk DNA paradigm is so important is because your best arguments for universal common ancestry, that kind of ancestry that says whales and strawberries are related, involves arguments that assume these DNA elements found in the genomes of living organisms are evolutionary leftovers. That's why the arguments they look to are pseudogenes. Pseudogenes, they argue, are genetic mistakes, basically, that are shared across the biological world. For example, they'll look to what they call pseudogenes that are shared in the same genetic loci or locations between humans and chimpanzees and argue that we must have inherited those genetic mistakes from a past common ancestor. Or they look to these regions of the genome that comprise retrotransposons, or what they call viral DNA. They assume this viral DNA got there through the ancient invasion of past viral infections. It's why they argue a lot of the genome is unconstrained or not conserved because it is mostly functionless. It's funny, we were talking about hierarchies earlier, and if evolution were true, we should find no homology. We should find very little in terms of a signature, a hierarchical signature, because think about it for a moment. If we've been evolving for millions of years and sequences in the genome that they say are non-functional evolutionary leftovers, for example, most serious evolutionists, they will argue that the genome is only about 10% functional, with the rest being non-functional or having no evidence for function. They argue a lot of those regions are unconstrained, as in they're not subject to selection. And so they say that these regions can accumulate mutations neutrally because selection is not subject to those genetic sequences. Well, wait a minute. If that's true, and these unconstrained or these sequences not being conserved have been mutating unchecked for millions of years, that should scramble any real signature. That should remove the hierarchical patterns that we observe. But the fact that we do observe hierarchies in unconstrained regions, or just constrained and unconstrained regions in general, tells us that the genome is young, and it is supporting evidence for the separate ancestry model. Because when God created the original created kinds with internal diversity, and this internal diversity can be utilized by natural selection, recombination, for example, the fact that you get new chromosomal combinations every single generation. With these DNA differences built in from the start, you can get new varieties quickly. You can get new varieties in a single generation. Well, that demands hierarchical patterns, because God would have created humans with pre-existing differences. God would have created felids and canids and fish and bears with pre-existing differences. And that's how we can explain the origin of species after the ark. After the ark, you have a bear archetype, or you have a felid archetype, or a canid archetype. From those pre-existing functional differences and the mechanisms of speciation that even evolutionists mostly hold to, including migration, isolation, inbreeding, genetic processes such as recombination, gene conversion, natural selection, mutations. You can have everything from a wolf to a coyote to your domestic dogs to your dingoes in terms of your felids. You can have everything from a tiger to a lion to a house cat to a jaguar to a cheetah, all because those DNA differences are built in from the start. They're front loaded. Pre-existing DNA diversity. In technical terms, this is called heterozygosity. This goes back to basic genetics. Think of alleles. Capital A, lowercase a, capital B, lowercase b. You'd be genetically heterozygous at that genetic location. But if you're capital A, capital A, capital B, you're genetically homozygous. There's no DNA diversity there. God commanded us to be fruitful and to multiply. It wouldn't make any sense for God to say, be fruitful and multiply, and then for that to be carried out through cloning. Well, that's exactly what you would get if God created Adam and Eve as homozygous clones. No, it makes sense both theologically and scientifically that God would have created Adam and Eve. Again, this applies universally across the biological world, with pre-existing DNA diversity, with internal diversity that can be manifested as change. There is a great deal of morphological adaptability built into every single kind. And over time, creatures, they hit walls, because there's only so many shifts in heterozygosity, which just means more allelic variability, to homozygosity, which means less allelic variability. And so eventually, let's take dogs, for example, you get something like a Chihuahua that has very little allelic variability. And so over time, creatures hit walls, and so there are limits to the change that is the potential for change that exists in living organisms. Okay, let's continue here. We were just talking about the junk DNA paradigm. And so if the junk DNA paradigm is overturned, and we can demonstrate that the majority of these DNA sequences, these DNA elements, are functional rather than non-functional and junk, then we can argue that they are there for a design purpose. And so this is the designed archetype model, where God creates original created archetypes. Those archetypes we find on the ark as well. Okay, roughly 2,000 years after the flood. And those DNA sequences and those DNA elements are there for a functional reason. By overturning the junk DNA paradigm, you overturn some of the so-called best lines of evidence for common descent. That includes ERVs, ALU sequences, pseudogenes. That also goes for the hierarchical patterns that they followed into. And that's why the evolutionary community has really struggled hard with the fact that we have strong evidence for genome-wide activity. The ENCODE project revealed, they uncovered 80% biochemical activity in the human genome. We know there's at least evidence that 60% of the genome is transcribed into RNA. Remember, we got DNA transcribed into RNA, translated into protein. And so over 60% of our genome is transcribed into RNA. The genome's producing these RNA transcripts. But the evolutionists want to say that this activity is spurious or it's just noise. It's not really important. But why in the world would the genome comprise so many sequences that are simply evolutionary leftovers? All of this activity is noise, when that would be wasteful of energy and resources to replicate all of this genetic content, to transcribe all of this DNA. There should be mechanisms over time, if evolution is true, that would weed out all of this junk, that would weed out or remove all of these evolutionary leftovers from the genome, because it is wasteful of energy and resources for the cell. It takes a lot of energy for transcription to take place. Every time you add a single nucleotide to a growing RNA chain, you are consuming a lot of energy. You are consuming ATP. And so again, if this really was just noise, there should be mechanisms within the genome to silence it. Natural selection over time should weed out this junk. Here's the thing. In order to really discover which parts of the genome are functional and whether or not this activity is beneficial activity, we have to study interactions between the systems of the cell. And when we do that, we find a lot of activity. And so if there are interactions taking place, which there is function. We have to look to transcription. We have to look to transcription factor binding. Okay, because we have in the genome, we have these important proteins. They're called transcription factors. And what they do is they regulate the transcription of genes. And so when DNA is copied into RNA, okay, DNA flows into RNA and RNA is translated into a protein. We have these transcription factors that are binding to sites in the genome to help initiate transcription. And these transcription factors, they assist in making sure basically that the right genes are being expressed in the right cells of the body at the right time. And so there's a lot of control, there's a lot of regulation going on in the genome. We also know that RNA polymerase attaches to the promoter, right? These genetic switches in the genome that initiates transcription, but only with the help of these proteins, these transcription factors. What that means is if we have high levels of transcription factor binding, then this should mean that this binding is functional binding, because we understand what transcription factors are there for. We understand what they're doing. And so if they're binding to places within the genome, but they're binding for no good reason, it's just random binding or non-specific binding, then what you are doing is gumming up systems within the cell. You're gumming up the operations. That's why most of the binding must be serving a purpose, or else cell operations would be very messy. Again, this is an energy-intensive process. And if these operations that we see taking place in the cell was mostly noise, this would be incredibly wasteful of energy and resources for the cell. there also wouldn't be enough transcription factors to go around to actually do the binding that is necessary for the cell's function. And so, yes, if we see a lot of binding, then it should be there for functional reasons. And that's why we have to look to transcription factor binding, protein binding to DNA, histone binding, three-dimensional interactions within the cell, histone modifications, RNA binding. These are all biochemical assays. And if there are interactions that are taking place, and these interactions actually have consequences or outcomes, then the most parsimonious explanation for this is function. And this is why the evolutionary community is really struggling with the evidence that suggests the human genome is 80% biochemically active. Because if the majority of the genome is functional, if it is active, then it overturns arguments like ERVs, and it makes deleterious mutation accumulation an even larger problem for evolutionists. We are just beginning to understand the DNA language. Every day we are understanding more and more about what's going on a molecular level. We've also got this amazing world of epigenetics that the evolutionists have a real difficult time explaining from a naturalistic perspective, because epigenetics has to do with adaptability. As God's created organisms navigate through different environments, genes within the organisms can actually be turned on or turned off. There's millions of DNA switches in biological organisms that are just waiting to be turned on or turned off through the right environmental stressor or cue. And it's all dependent upon what is necessary to survive the environment. And so epigenetics basically results in a change within the activity of the genes. But it is a change that is above or beyond the DNA itself. This is amazing evidence of forward thinking, which, as I like to say, points us back to the forward thinker. And so you're going to see throughout this book a great deal of time spent refuting junk DNA in general. And so let's continue here. I spend most of chapter 3 going over the non-discriminatory lines of evidence that relate to the creation versus evolution debate. I say most because I do spend the final portion of chapter 3 covering some of the amazing lines of evidence that apologists of evolution are incapable of addressing from their position. Everything leading up to the must-read chapter 4 of this comprehensive handbook has been strategically constructed and laid out in a way that fully sets the stage for what the reader will be presented with is in what is undoubtedly the most exhaustive chapter found in this book on endogenous retroviruses. And that wraps up the introduction chapter. I do want to provide just a little bit more commentary, and I think this will help really set the stage for what's to come in part 3, where we really get into the meat of the book, the chapters themselves. So you'll find in part 3, or chapter 1 technically, the important details. What is a retrovirus? What's the difference between an exogenous retrovirus like HIV, that's the retrovirus that causes AIDS, and an endogenous retroviruses occurring from within. Endogenous retroviruses result from vertical transmission, and an exogenous retrovirus infects horizontally, like HIV, from person to person. When it comes to HIV, for example, the HIV genetic material is incorporated into the host genome, because retroviruses have the capability of performing what's called reverse transcription. What is reverse transcription? Well, it's just transcription in reverse. So you got DNA to RNA to protein. So reverse transcription is RNA to DNA. So a retrovirus is made of RNA genetic material, and that RNA genetic material is reverse transcribed into DNA. And during these instances where the DNA that has been reverse transcribed from the retroviral material can actually be incorporated into the host genome through the activity of a very specific enzyme called integrase. If it ends in ACE, then you can be sure that we're looking at an enzyme. Integrase, reverse transcriptase, protease, and this specific process is called endogenization. And that is when the retrovirus is incorporated into the genome, where it can exist in latent form, or it can be transcribed to produce RNA molecules, which can reactivate the infection. Okay. But a way that retrotransposons can actually proliferate throughout the genome is through a very interesting, a very complicated mechanism called retrotransposition. You can have multiple copies of an ERV that can become proliferated through the genome of an individual. And when retroviral DNA is transcribed, you have the GAG that we talked about, the pol and the pro. These are the components of the ERV. You've also got the LTRs on either side, the long terminal repeats. You've also got the ENV gene. They will produce polyproteins that are cut apart by protease into individual proteins to carry out their individual activities. Now, a more expansive explanation of what a retrovirus is and what's going on in terms of endogenization will be provided in part 3. But I do want to focus on the several assumptions that the evolutionists make as they interpret ERVs as being evidence for common descent, that we are going to deal with extensively throughout this series, but also as I have in my book. One, they assume ERVs are non-functional. 2, that the insertion mechanism is random. And 3, that the mutational events taking place are also random. Because we have mounting evidence that the retrotranspositioning that I just talked about of these retroviral elements is not necessarily a random process, but actually a non-random process, where certain sequences serve preferentially. But another issue involves the solo LTRs. We talked about this at the beginning of the part of this episode, because these solo LTRs, they comprise about 90% of what evolutionists call endogenous retroviruses. They're found in host genomes as functional elements. I talked about in part 1 how they act as a regulator to control gene expression, which is the process that turns a gene on in the cell to make RNA and proteins. And transcription factors, which we talked about earlier, those proteins that regulate the transcription of genes, they bind to specific parts of the DNA to make sure that DNA is being copied into RNA, which is, again, transcription. They also help to ensure that the right genes are being expressed in the right cells of the body at the right time. There's so much control. There's so much regulation going on in the genome. It is hard to believe that anybody could think that this happened by chance for no reason at all. This is some complicated stuff. Now, the solo LTRs, in light of their functional roles, since they are serving in the genome and serving in a way where they act in gene regulation, they can function as promoters, and again, they can serve as transcription factor binding sites. Now, if we look at the structure of the retroviral material, the LTRs serve as promoter sequences and serve as binding sites for RNA polymerase. It's critical for transcription. Now, if you were a designer, let's say, you could incorporate similarities to the long terminal repeat to genomes of biological organisms. You could do this to genomes of eukaryotic organisms, for example, because they have elements or properties that make them ideally suited for what? Promoter sequences. So you can use these elements that, yes, they are elements that are found as components in retroviruses, but because of their properties, they can be well suited for other functional sequences like promoter sequences. And the fact that we see these LTRs that are simply isolated, because remember, when an evolutionist says, oh, we share so many ERV sequences with the primates, what they're not telling you is it's not that we share full ERVs where they comprise LTR, GAG, POL, ENV, LTR. Remember, because the LTRs are on either side. No, it's that we find a few like that, but over 90% of so-called ERV sequences are, again, solo LTRs. And they're found in isolation and associated with host genes. Why are we to believe or assume the evolutionary story is true that these are degenerated ERVs? They are better explained as functional stretches of DNA. And so the reason we find so many thousands of these sequences shared in the biological world is for, again, design purposes. Now, they do share design features with the promoter sequences of retroviruses. But because they have those specific design properties to carry out that useful role, then it should be intuitive that they're there because they were intentionally incorporated, since they consist of very specific design properties that make them useful in particular settings. So in the genome, acting as promoters, but then also in a retrovirus, acting as their own functional role. Now, the reason why I say this, because one of the main criticisms that has been brought forth, and I'm going to address this in detail in future presentations, Dr. Zach Hancock, he's a population geneticist, and he basically asked rhetorically, why would God use these solo LTRs that have similar properties or the same properties to LTRs in retroviruses? Why would he need to use thousands and thousands of basically pieces of retroviruses to help with genomic function? Well, again, that is answered by the fact that these design features, they comprise very specific design properties that make them useful in not just one setting, but in multiple settings. the setting of a retrovirus or the setting of an isolated DNA element that is functioning in a way that's useful for the cell. Now, evolutionists, they have to argue that the long terminal repeats, the reason why they exist in isolation is because they got disassociated from the endogenous retrovirus and ended up being associated with a particular host cell gene. But it's funny because this association has to be at just the right integral position in order to function as a promoter, and that it is going to alter the gene expression profile of that gene. Now, that is far-fetched evolutionary storytelling. This will end up having biological consequences for the organism. And so, as with a lot of things, evolutionists have to argue for the right set of events to incorporate an LTR from the ERV because it's been disassociated from the ERV, right? And insert it in the perfect position to just coincidentally serve as a promoter that changes in the gene expression profile are the just right changes to ensure survivability or ensure a benefit to the host organism. Again, this is based on storytelling. The evolutionist, and it's not just, well, a couple times that this has to happen according to the evolutionary model. No, we have evidence that thousands and thousands of these are functioning the way they do. And so for sake of argument, let's say to the evolutionists, sure, maybe this coincidentally happened a handful of times. But to happen so frequently, to happen routinely, and to be widespread, this pushes the boundaries of reality. This is pseudoscience here that the evolutionists are pushing. Just because the LTRs of retroviral sequences are similar to the solo LTRs of organismal DNA is not compelling evidence for common descent. And this is a main criticism advanced by Dr. Zach Hancock, and I do appreciate his examination of my work on endogenous retroviruses. And I plan to put all this out in the form of a presentation at some point in the future. I just want to provide the listeners with some beneficial and worthwhile feedback as we work towards model building and work at refuting all objections and criticisms from the skeptics. It makes far better sense from a design perspective that these solo LTRs that are shared between all forms of life are designed stretches of DNA and therefore exist for design purposes. And so I think we're gonna wrap it up there. We've basically reached the time mark that we reached in part 1. We've gone about 55 minutes. And so that concludes part 2. If you haven't yet seen part 1, please do go check it out because I provided a lot of worthwhile commentary there as well. Stay tuned for part 3. Part 3 is going to be chapter 1, the important details. And this chapter really sets the foundation for what will be discussed in the rest of the book. And I promise you, it is going to be comprehensive. We are dismantling, we are refuting the very best evidence for common descent. God bless all.